For decades, if you opened up a standard medical textbook and looked up cancer, you were usually presented with this incredibly static image. It
Absolutely.
was depicted almost like, I don't know, an architectural defect, just a rogue lump of mutant cells sitting in a specific organ waiting to be surgically removed.
Right, which is a very reassuring way to look at a terrifying problem, honestly.
Right.
I mean, we like our diseases to be categorical and contained.
Yeah.
You find the lump, you cut it out, or you radiate that specific geographic area, and supposedly the problem is solved.
But then you step out of the anatomy lab and into the actual molecular biology of modern oncology, and you realize that static textbook diagram is just, well, it's entirely wrong.
It's completely wrong.
We aren't dealing with a lump. We are looking at a biological landscape that is incredibly dynamic. It is a hostile, living entity.
It is the absolute definition of an evolving pseudo-organ.
A pseudo-organ.
Exactly. And that's really the paradigm shift we are exploring today. A tumor doesn't just sit passively in the tissue. It actively parasitizes the host.
Right.
It hotwires your body's most complex natural systems, your metabolism, your blood supply, your immune system, all to guarantee its own survival.
And that brings us to the core of today's deep dive. We are exploring the Handbook of Targeted Cancer Therapy and Immunotherapy, the second edition, by Doctors Daniel Karp and Gerald Falchuk.
It's an incredible resource.
It really is. And whether you are a medical student navigating your clinical rotations, a young healthcare professional, or just someone fiercely dedicated to understanding the cutting edge of science, we are going to unpack how this pseudo-organ actually operates. And more importantly, how modern medicine is finally learning to outsmart it. We aren't just looking at what cancer is, we're looking at how it behaves.
And that behavior, it starts at the most fundamental level, which is energy.
Okay, let's unpack this.
Right, so if a tumor is going to grow from a single mutated cell into a massive, hostile pseudo-organ, it requires a staggering amount of resources. It has to completely rewire its own cellular metabolism.
Right, and to understand how bizarre this metabolic rewiring is, we have to look at how a normal, healthy cell gets its energy. It's an incredibly elegant, highly efficient system, isn't it?
It is. So in a healthy cell, glucose sugar enters the cytoplasm, and through a process called glycolysis, that glucose is broken down into pyruvic acid.
Which yields a tiny bit of energy.
It's just a tiny bit, yeah. But then that pyruvic acid is sent into the cell's power plant, the mitochondria.
Right.
And in the presence of oxygen, the mitochondria perform oxidative phosphorylation. It is a brilliant piece of biological machinery that squeezes every last drop of energy out of that glucose molecule.
Producing roughly, 36 molecules of ATP?
Exactly, 36 ATP, which is the basic energy currency of the cell.
So it's like a highly efficient hybrid car engine. You put a little bit of fuel in and you get a massive amount of mileage out of it.
Precisely. But way back in the 1930s, a scientist named Otto Warburg noticed something that defied all biological logic.
Oh, wow, the 1930s.
Yeah, a long time ago. He observed that cancer cells, even when they are surrounded by plenty of oxygen, completely shut down that highly efficient mitochondrial power plant.
They just turn it off.
They do. They rely almost entirely on that first primitive step, the glycolysis.
Which makes zero sense at first glance. I mean, by skipping the mitochondria, the cancer cell goes from making 36 ATP molecules down to just 2.
Right.
It is an incredibly inefficient way to generate power. Going back to the car analogy, it's like ripping out that highly efficient hybrid engine and dropping in a massive gas-guzzling V8. You completely sacrifice fuel efficiency.
You do. But consider what that V8 engine gives you in return.
Pure explosive speed.
Exactly. What's fascinating here is that what we now call the Warburg effect isn't a mistake. It's a calculated biological trade-off. Cancer cells don't just need pure energy.
Right, they're dividing.
They are dividing so rapidly that they desperately need physical building blocks. They need carbon, nitrogen, lipids, and amino acids to actually create the physical structure of new daughter cells.
Ah, so if it burns the glucose completely for energy, there's nothing left over to build with.
Exactly.
Yeah.
By stopping the breakdown of glucose halfway through the process, the cancer cell leaves a massive surplus of metabolic intermediates just floating around in its cytoplasm.
Wow.
It essentially hoards these half-digested sugar molecules and repurposes them as the raw lumber and bricks needed to build new cells. It sacrifices energy efficiency to become a hyperactive factory for physical mass.
That is brilliantly insidious. But obviously, to run a massive, inefficient factory like that, you need a ridiculous amount of raw material. Like, you need a constant flood of glucose.
Which is a massive vulnerability.
Right.
From a clinical perspective, if we know a tumor is hoarding glucose at 10 or 20 times the rate of a normal cell, we can target that specific metabolic pathway.
Exploit the weakness.
Right. We can design therapies that exploit that localized hunger, essentially starving the tumor supply lines without harming the healthy cells that are still using their standard efficient mitochondria.
But this factory doesn't exist in a vacuum. Cancer doesn't just change its own internal chemistry, it completely alters its surrounding neighborhood.
Oh, heavily.
Because if it's growing that fast and consuming that much sugar, it's going to run out of local supplies very quickly.
It does. A tumor can only grow to about 1 or 2 millimeters in size before it maxes out the local nutrient supply.
That's tiny.
Yeah, it's microscopic. At that point, the cells in the center of the mass become hypoxic. They are literally suffocating from a lack of oxygen.
So if the tumor doesn't adapt, it dies.
Exactly. So it flips what we call the angiogenic switch.
Angiogenesis. So the creation of new blood vessels, it basically forces the body to build new roads directly to the tumor. How does it manage that?
It sends out a chemical distress signal. The most well-known of these is vascular endothelial growth factor, or VEGF.
VEG.
Right. In a healthy body, there is a very delicate balance between signals that tell blood vessels to grow and signals that tell them to stop.
Sure.
But the suffocating tumor pumps out massive, overwhelming amounts of VEGF. This chemical bathes the nearby healthy blood vessels and literally forces them to sprout new capillaries.
But these aren't normal healthy blood vessels, are they? Because the signal is so chaotic and urgent, the infrastructure has to be terrible.
They're completely dysfunctional. The new blood vessels are chaotic, disorganized, highly leaky.
Leaky.
Yeah, structurally very weak. But they serve their purpose. They route fresh blood, oxygen, and glucose directly into the heart of the tumor, feeding that metabolic factory we just talked about.
It's literally hijacking our own plumbing. And the source material points out that the tumor goes even further. It constructs what's called a microenvironment.
Yes, the trifecta.
So the tumor is basically acting like a mob boss.
I like that analogy.
Right. It isn't just building an illegal factory, it's recruiting local, innocent bystanders to help. It bribes nearby structural cells, the fibroblasts, to build a thick physical scar-like shield around the tumor to protect it.
The microenvironment is perhaps the most heavily researched area in modern oncology for exactly that reason. But the mob boss analogy goes even deeper. Because what does a mob boss do when the authorities show up?
They bribe the police.
Precisely. Your immune system is the police force. Normally, specialized white blood cells like cytotoxic T cells are constantly patrolling your tissue.
Or in for threats.
Right. When they see a mutated cancerous cell, they're designed to lock onto it and destroy it. But the tumor deploys chemical countermeasures. It secretes specific proteins like TGF-beta into its neighborhood that literally paralyze those attacking T cells.
It puts the patrol officers to sleep.
And then it actively recruits corrupt officers. Your immune system naturally contains cells whose entire job is to suppress inflammation, to calm things down so you don't develop an autoimmune disease.
Like regulatory T cells and MDSCs.
Exactly. Tregs and myeloid-derived suppressor cells. The tumor chemically attracts these suppressive cells, pulling them into its microenvironment and uses them as bodyguards.
Wow.
So if a healthy attacking T cell tries to breach the perimeter, the tumor's recruited bodyguards will neutralize it.
Which completely explains why simply blasting a patient with traditional systemic chemotherapy often fails.
Yes.
You are trying to carpet bomb a bunker that is actively protected by the patient's own biological shields.
Yeah.
But this is where targeted therapy steps in. We can cut the supply lines.
We can. Take that distress signal we mentioned earlier, VEGF. Modern medicine has developed monoclonal antibodies like bevacizumab, which you might know as Avastin.
Right.
This drug is engineered in a laboratory to circulate through the bloodstream, find that specific VEGF distress signal, and bind to it, neutralizing it before it can reach the healthy blood vessels.
So the message never gets delivered. The new blood vessels don't sprout, and the tumor is choked off from its supply of glucose and oxygen.
Exactly. But, cutting off the blood supply is only half the battle. If the tumor is using our own immune system as a bodyguard and chemically camouflaging itself, we have to figure out how to unmask it.
We have to turn the immune system back into an attacker.
Exactly.
And here's where it gets really interesting. We are talking about the immunotherapy revolution, specifically checkpoint blockade.
Yes.
You mentioned earlier that the tumor puts the attacking T cells to sleep. How exactly does a tumor pull that off on a molecular level?
It exploits a natural safety mechanism. T cells are incredibly destructive, so they have built-in molecular brakes, known as immune checkpoints. One of the primary brakes is a receptor on the T cell called PD-1.
PD-1.
Right. Think of it as an off switch. When a patrolling T cell bumps into a normal, healthy lung cell or heart cell, the healthy cell reaches out and engages that PD-1 receptor. It's like a molecular handshake that says, hey, I belong here. I'm a healthy part of the body. Do not attack me.
It's a biological friend or foe identification system.
Exactly. Without it, your immune system would constantly attack your own organs. But cancer cells are devious. They evolved to heavily express the exact same handshake protein on their surface.
Of course they do.
So when the T cell comes up to inspect the mutant cancer cell reaches out, hits the PD-1 off switch, and the T cell is instantly deactivated. The cancer walks right past the security checkpoint.
It's a perfect molecular disguise. But researchers realized, what if we just block the handshake?
That was the breakthrough that won the Nobel Prize. We developed targeted drugs, again, monoclonal antibodies like pembrolizumab and nivolumab, that physically cap that off switch.
They just cover it up.
Yeah, they bind to the PD-1 receptor on the T cell so the cancer cell can't reach it.
You literally tape over the off switch.
You remove the brakes, T cell bumps into the cancer tries to hit the off switch, but it's blocked. The T cell recognizes the mutant for what it is, sounds the alarm, and destroys it.
And this mechanism led to one of the most profound paradigm shifts covered in our source material. In 2017, the FDA approved pembrolizumab for a very specific type of patient, and it completely changed how we categorize the disease.
It really did.
They approved it for any advanced solid tumor that has a specific genetic defect called microsatellite instability high, or MSI high.
The term for this is organ-agnostic therapy, and you cannot overstate how revolutionary it is.
Because for the entire history of medicine, we categorize cancer by its zip code. You had lung cancer, or you had colon cancer, or breast cancer, but this approval said the zip code doesn't matter anymore.
It really doesn't. If a tumor is MSI high, it means its DNA repair mechanisms are completely broken. It is accumulating hundreds and hundreds of random mutations.
So it's very messy.
Highly chaotic. And to the immune system, a cell with that much genetic trash inside it looks incredibly foreign. It is highly visible.
The only reason the immune system hasn't destroyed it is because the tumor is hammering that PD-1 off switch.
Correct. So whether that tumor is sitting in the liver, the pancreas, or the skin, if it has that specific MSI high fingerprint, we know that blocking the PD-1 off switch will allow the immune system to see all those mutations and clear it out.
We are treating the molecular mechanism, not the anatomy.
Exactly.
Okay, but I have to push back here. You are systematically removing the molecular brakes from a patient's immune system.
Yes.
You're disabling the very safety mechanism that prevents the white blood cells from destroying healthy tissue. There has to be a catch.
Oh, there is a significant catch. And if we connect this to the bigger picture, it's a double-edged sword of immunotherapy. When you take the brakes off the immune system, you are inviting autoimmunity.
Right.
The T cells wake up and attack the cancer, but they can also become hyperactive and start attacking the patient's healthy organs.
The literature calls these immune-related adverse events.
Yes. And for the medical professionals listening to this, recognizing and managing these toxicities is now an absolutely critical daily skill. The era of just managing chemotherapy-induced nausea and hair loss is over.
It's a whole new ballgame.
It is. You are now looking for immune cells infiltrating the lungs and causing severe pneumonitis. You are monitoring liver enzymes for immune-induced hepatitis. You are watching for the immune system completely destroying the thyroid gland.
So what do you do? I mean, if a patient comes into the clinic and their T cells are actively destroying their liver because of the immunotherapy drug, you can't just tell them to push through it.
Absolutely not. The clinical guidelines are very strict on this. If a patient develops a severe grade 3 or 4 immune-related toxicity, you halt the immunotherapy immediately.
Okay.
You admit them to the hospital, and you deploy high-dose intravenous corticosteroids. You basically use powerful anti-inflammatory drugs to forcefully suppress the immune system back down to a safe baseline, putting out the fire before it destroys the organ.
So you are literally walking a tightrope. You want the immune system angry enough to kill the cancer, but not so angry that it kills the host.
And it requires extreme vigilance. But when it works, you achieve durable, long-lasting remissions that were medically impossible 20 years ago.
That's incredible. But let's look at the flip side of targeted therapy, because we established early on that cancer is a nimble, evolving pseudo-organ. It adapts. We hit it with these incredibly smart, hyper-specific pharmaceutical weapons, and it just mutates to evade them.
This is the immense frustration of modern oncology. The human genome encodes over 500 different kinases. These are the cellular enzymes that act as signaling switches for growth and division.
Right.
Cancer cells are brilliantly chaotic. If you block one signaling pathway with a drug, the cancer will eventually mutate and find a workaround using a different kinase.
Which makes it sound like we were just playing an endless multi-billion-dollar game of whack-a-mole.
In many ways, we are. Consider patients with non-small cell lung cancer, whose tumors are driven by a specific mutation in the epidermal growth factor receptor, or EGFR.
Okay.
The receptor is stuck in the on position, telling the cell to divide endlessly.
So researchers designed a pill, a first-generation targeted therapy that perfectly fits into that broken receptor and shuts it off.
And the initial clinical response is often miraculous. The patient takes a pill, the signaling stops, and massive tumors melt away in weeks.
Boom.
But hidden inside that dying tumor are a few clonogenic stem cells.
The cancer's backup files.
Exactly.
Yeah.
And because they are genetically unstable, they undergo serial mutations. Over months or years, one of those surviving cells develops a very specific resistance mutation. The most famous in this context is the T790M mutation.
What does that actually mean for the drug, though?
It means the physical, three-dimensional shape of the EGFR receptor literally changes. The binding pocket alters just enough that the first-generation pill physically cannot fit into it anymore.
Wow. The lock has been changed.
Exactly. The lock has been changed. The cancer roars back, and it is completely immune to the drug that just saved the patient's life a year prior.
That is devastating.
It is, but the science doesn't stop.
Yeah.
Because researchers were able to biopsy that resistant tumor, sequence its DNA, and identify that exact T790M shape change. They went back to the lab.
They made a new key.
Right. They designed third-generation targeted drugs like osimertinib, built specifically to fit the new mutated lock. And it works brilliantly. We see a 61% response rate in trials.
But eventually, it's going to mutate again. The whack-a-mole game continues. If the human genome encodes over 500 kinases, and cancer can continually mutate to find a new pathway, aren't we just throwing static chemical pills at a living, thinking enemy? Eventually, we run out of pills. What happens then?
Well, when static drugs fail, we have to escalate. If the cancer is a living, evolving entity, we need a living, evolving drug to hunt it down.
You're talking about CAR-RT cell therapy.
Yes.
Chimeric antigen receptor T cells. Every time I read about this, it feels like science fiction that somehow slipped into clinical reality. Let's walk through the actual engineering process detailed in the source material because it is mind-blowing.
It really is.
Let's look at a patient with aggressive acute lymphoblastic leukemia, or ALL, whose cancer has outsmarted every pill and every chemo drip we have. We start by literally extracting their blood, right?
We do. Through a process called apheresis, the patient is hooked up to a machine that filters their blood and pulls out their native T cells.
Okay.
At this moment, these T cells are useless. They are completely blind to the leukemia because the cancer has camouflaged itself so well. We take those blind T cells and transport them to a highly specialized cellular engineering laboratory.
This is where the hacking happens. We have to reprogram their DNA.
Right. We use a retroviral vector, essentially a hollowed-out, deactivated virus that can no longer cause disease, but still retains its ability to inject genetic code into a host cell.
That's wild.
We use that virus to insert a brand new set of instructions into the patient's T cell DNA. We force the cell to build a synthetic artificial receptor on its surface.
The chimeric antigen receptor.
Yes, chimeric meaning it is stitched together from different biological parts. The receptor has two crucial components. On the outside of the T cell, we engineer a homing beacon. It is perfectly designed to latch onto a very specific protein found only on the surface of the leukemia cells, usually an antigen called CD19.
Okay, so now the T cell has molecular night vision goggles. It can see the CD19 protein on the cancer cell, and it physically grabs it. But seeing the enemy isn't enough, right? It has to know to pull the trigger.
Which brings us to the inside of the cell. The internal portion of this artificial receptor is wired directly into the T cell's activation machinery. We use signaling domains like the CD3 zeta chain.
What does that do?
When the outside of the receptor grabs the leukemia cell, the inside of the receptor sends a massive, explosive electrical and chemical signal into the T cell. It screams at the cell to activate, to clone itself thousands of times over, and to instantly destroy whatever it just grabbed.
We take a few of these hacked cells, grow millions of them in bioreactors, and then infuse this customized army back into the patient's bloodstream.
Like Tzinlakuso.
Right. It is a living drug. It actively hunts, it multiplies when it finds the target, and it persists in the body to guard against a relapse.
And the clinical results for patients who previously had a 0% chance of survival are staggering.
Yeah.
We are seeing complete, durable remissions in highly refractory leukemias and lymphomas. But, as with everything we've discussed today, when you manipulate biology at this level, the blowback can be catastrophic.
What happens when millions of highly aggressive, genetically engineered super soldiers hit a massive tumor burden all at once?
It creates a biological explosion. As the CAR-T cells slaughter the leukemia, they release a massive wave of inflammatory distress signals into the bloodstream. This is called cytokine release syndrome, or CRS.
The immune system basically overloads the host's circuitry.
It is a systemic inflammatory storm. The patient can spike dangerously high fevers, their blood pressure can bottom out, their lungs can fill with fluid, and they can experience severe neurological toxicity, hallucinations, tremors, even comas.
That's terrifying.
Managing a patient through a severe CRS episode requires a highly coordinated intensive care team. We often have to use yet another targeted antibody called tocilizumab just to block the specific inflammatory molecules causing the storm, hoping to keep the patient alive while the CRT cells finish the job.
It is the ultimate high-wire act in medicine, which really brings the entire journey of this deep dive into focus. We started by looking at a static textbook picture, and we've ended up in an intensive care unit managing genetically engineered cellular armies.
That is the reality of modern targeted therapy. We've traced how cancer initiates its survival by hijacking cellular metabolism through the Warburg effect. We explored how it builds its own infrastructure and bribes the immune system to create a suppressive microenvironment.
Right.
We looked at how we are stripping away those disguises with checkpoint inhibitors, and finally, how we are countering its endless mutations by rewriting the very code of the immune system.
So, what does this all mean for you? If you are studying for your boards or you are treating patients in the clinic tomorrow morning, the ultimate takeaway from this material is that your understanding of this disease must continually adapt, just as the disease itself adapts. You cannot view it as a localized, static event. It is a wildly complex systemic war.
And the deeper we look into the science, the more interconnected that systemic war becomes.
Which perfectly sets up the final thought we want to leave you with today. Before we wrap up, there is a phenomenon buried in the clinical history of this research that sounds less like biology and more like outright magic.
Oh, this is a fascinating puzzle.
It really is. It's a mystery that forces us to rethink everything we know about localized treatment. We discussed how localized treatments affect local tumors, but historically, physicians have occasionally noted a bizarre systemic reaction in patients with widely metastatic disease.
Right. Imagine a patient who has a primary melanoma tumor on their left arm, but the cancer has already spread, creating distant metastatic tumors in their right leg and their lungs.
The physician decides to apply targeted, localized radiation only to the primary tumor on the left arm. They don't touch the tumors in the leg or the lungs. They are just trying to control the local pain.
But then something impossible happens. A few weeks after radiating the arm, the distant tumors in the leg and the lungs suddenly begin to shrink, and in rare cases, they completely disappear.
Wait, really? You radiate the left arm and a tumor on the right leg melts away. It defies the localized model of medicine.
It completely shatters it.
So, based on everything we have unpacked today regarding the immune system, tumor microenvironments, and how cancer hides, here is a review question and a thought experiment for you to mull over. What is this mind-bending phenomenon called, and what does it suggest about the relationship between localized cell death and systemic immune response?
I highly encourage you to dig into the research on this. It's known as the abscopal effect. Consider what might be happening on a microscopic level. When that localized radiation destroys the primary tumor, it ruptures the cells, spilling massive amounts of mutated proteins, hidden cancer antigens, into the bloodstream.
So think about how the immune system, which was previously blind to the cancer, might react to suddenly being flooded with this biological debris.
Exactly.
It's acting like an accidental in vivo vaccine. The radiation doesn't just kill the local tumor, it accidentally trains the immune system to recognize the enemy and sends it out to hunt down the rest of the cancer globally.
It is a profound realization. It proves that every intervention we make reverberates through the entire biological system.
It changes how we view the whole battlefield. That murky X-ray isn't just a defect, it's a dynamic systemic conflict. And as the Handbook of Targeted Cancer Therapy and Immunotherapy proves, we are finally building the weapons to win it. Thank you for joining us on this rigorous exploration. Keep learning, keep questioning, and keep diving deep into the science.